So, welcome you all in this fifth webinar series of IBDC. And in this webinar series, I will discuss this Proteome data portals, which is hosted at IBDC. And during this webinar, I will discuss more towards this submission protocols, standards, and file formats, and a short demo about this IPD web portals. And as a general instructions, this microphone will be muted. If you have any question, you can raise via this chat box. And if you, in any case, you miss this session or webinar, you can check back in the recorded session, which is available at the IBDC website. So start with this biological data types. So as we know, the biological data types start at the initial level, start with this gene, genome, and variation with the genetic content to functional profile, the functional expression, the functional genomics, and then which is converted to this proteomics part. Proteomics expression, proteomics, then convert this, move down to the 3D macromolecular entity which interact with itself or via the small molecule. So that is the small molecule metabolite study. And then now we are also there are certain type of biological images are also generating to use to the diagnosis of the disease. So the disease this image portals, and then the phenotypic traits to analyze the phenotypic data. So to capture all this information at the IBDC, we have different number of portals like INDA, which take the submission regarding this nucleotide data. Then this ICPD, which is the phenome related database. Then ISDA, which is the 3D macromolecular structure archive data. Then IMDA take the submission related to metabolome study. Then IPD take the submission related to the proteome study. And then very latest is the IBIA, which is image related data archives. So in this webinar, I will discuss the agenda of this webinar is to give the overview about the submission process in the IPD, then standard for the submitting proteomics data, data access protocols, and live demonstration about the how user can get the submit the data at the IPD portals. So start with the proteome. So proteome is the collection of compendium which is encoded by the genome, and this compendium includes all form of the isoforms and its modifications coming from the samples. So if you understand this, extract the protein from the samples, separate the proteins, then using the analytical approach, you can identify the protein identifiers, and using the computational workflow, you can characterize the protein type and function, and this protein profiling, etc. So, and if we see the role of the MS in the proteome. So MS take the biological samples as a source, then using this ionizer, it's ionize the samples, and then samples are analyzed by the analyzer using this time of flight mode, which include the M by G value, and then ion detector will capture all the ions, detect the ions, then data system will analyze all the data and produce the mass spectrum. So, and this further, this mass spectra can be further decoded to identify the background biological entity which is mapping to the spectra signature. And why it is used as a main source of protein identification? Because it's the method of choice for identification and characterization of the proteins. It provides high level of sensitivity, speed, and flexibility to adopt with different workflow type of the proteomics. It supports the high throughput nature to analyze the samples, and it also supports the different workflow to analyze the protein, like the discovery-based identification or the targeted identification. So different type of setup means it can be set up around the MS to analyze the proteomics data. And as this MS produce high volume of the data, so we need a systematic access so that user can get this data, deposit that data and access that data in a very systematic manner. If we combine both proteomics and MS, so general workflow is like extract the protein from the samples, then digest the proteins to this peptide part, and then these peptides are separated by some liquid chromatography, then ionizations, which ionization pass through the MS analyzer and the detector, so that this MS part identify the full spectra of this proteome. And this, if you want to further amplify the signature of this spectra, then you can further carry forward this signature to identify the MS spectra. And this spectra is very customizable and can be used to employ along with the different type of this proteome identification approach. And if we look at this application part of this proteome, so it is very well known for this pathogen diagnosis, then protein translation modification identification, and then protein mutation identification. And if you, the therapeutic part, if you look at then proteomics therapeutic target identification, given the drug type, the profiling of this protein within the samples, then pathological progress within the organism, this organ tissue, then identification and development of the biomarker, and course of development of the pathogenesis, and then also the given the drug type to identify the mechanism and the drug molecules from the proteome expressions. So where we stand means what is the role of the IPD? So IPD serves as a repository for facilitating this submission, management, and dissemination of this mass spectrometry data, which is generated across the lab and beyond, and in India and beyond. So, and IPD was developed as per the guideline from the Proteome Exchange data submission policies. You can check in this this link, and all the user need to fill the minimum required field for the easy submission. IPD also provide this complete and partial submission type that I will discuss in the following present this PPT, and then support it also support machine type generated raw file, peak list file, result files, along with the other necessary files. And user can submit your data either via the web or the FTP based submission, and depending upon the size of the data you are transferring from your side to IPD. After validation, each submission will receive a unique SSN number, which can, and this submitted data will be open access as per the embargo times. So these are the standard features that IPD follows to archiving and management of the proteomics data. So now this IPD is very vocal about this FAIR principle to support this FAIR principle. FAIR stands for this findability. That means all data should be associated with very standard and stable SSN, so that you can find your data in any time. Then accessibility, it should be very longly support to access your data in any point of time. And then interoperability to get your cross-reference, use the standard mechanism or the metadata fields and the ontology fields in such a way so that you can cross-reference your data fields to the other data repository. And then usability, that means if you are downloading this data, so you will have enough sufficient information so that reprocess or re-analyze your data at your site. So this IPD supports all these FAIR principles during this submission and management process. So if you access the IPD submission, user have to first register using the registration form and then log into the portal. After login to the portals, you will get to the IPD dashboards. Before going to the dashboard, I just show what sort of information user need to fill to a proper submission. So it's the first you have to create a form related to this project. So basic information about the project you have to fill, then experimental details, type of samples, study, and then separation method, characterization approach you have used to during the experimental setup, and then what are the background metadata information like the experimental type, disease, associated disease information, fluid information, and then organism information. Like that, you can fill in the metadata information, then move to the upload file so that the user can upload all the data related to the projects in the in your project, and then final submit the data. Once IPD got your submission, it will validate your all the process and then provide the assistance. After the embargo time, which is in general 1 year for open access, manage access this submission. So after embargo time, all data will be open access and user can browse the IPD archive, search collection using the different fields which is available in the IPD portals, then access project details, download the project and metadata files. So during this submission, user will be asked to provide the type of submissions, and this submission type is depends upon the type of the files and the files you are providing during the file submission. So if you are providing this 3 set of files which include the raw spectrum files, which is the machine generated files, binary files, then result process file, result identification in the mg-ident.ml or the mg-tab file, and then peak list file, that is the process spectra files in the mgml, mgxml or mgf file. Then it will be user can submit all this data in the complete submission type, because you are providing this result file. It can be further processed and associate your result data in the downstream manner. But if you are providing the partial submission, you only need to upload the this raw spectrum files and then search engine files. And optional files also include this database file. What are the background database you have used to identify this protein, and then spectral library you have used to map this spectral library, then quantification results if you have any, then gel image, method and protocols in the text format, and any other supplementary files image you can provide as a optional fields which are related to your projects. So this when you log into your dashboard, you will find a very interactive dashboard, which is not only provide the status about your project, how many projects you have created, and each stage how many information you have filled up, but also provide the progress, how much you have completed the ongoing projects. So as soon as you fill up the information about each detail on the heads, you will get updated about the status of the project. So in the dashboard level, you will get the information, summary information that are the created project information. During the this project creation stage, if you find you want to either update any entry, you can use the update button to update the project information. Then you can click over this highlighted this blue color entry to get the status and the information that you have already filled within the project. And if you have find any project which is not relevant to far the submission, but you have created that, then you can use the delete button to delete the project. So this is the in the user dashboard, user can facilitate this many information. At the current status, after inception during the July 2024, user space are spanning throughout the India, and at current we have around 26 registered users, which have submitted around 42 projects. And if we will look at the file level, so 374 search file, 1185 peak files, and more than 5,000 spectra files. And as I have already stated, there are two types of data access, which is the organic open access, which has come with the embargo time of 1 year. If you have part of any project which generate any sensitive data, or the you want to data which do not make it to open access, then you can write us as a request to submit your data as a control access. Data review committee will access your request, and based on their own result, you can access you can submit the data in under two hoods. At present, so this is this timeline growth of this data from the July 2024 to December 2024. So data is incrementally increasing in size and then number. So when the data goes under the open access, then user can browse all sort of data. And then in the browse table, you will get the information about the projects. And if you click over this project ID, you will get the each information about this project, affiliation, metadata information, experimental information, as well as you can download this information in the PDF report files. And in the downside, you will also find the listed associated files which you can file. You will see the file name type and then file name and the file type, then download button, so that you can easily download the file from the web interface. So we have to provide the real-time statistics about the data submission and open access data. You can check our visualization part, which include this how many disease type of information is submitted, how many cell type, tissue type, and then what are the status about the different file format which is submitted at present in the IPD. And if you hover at any data in bar chart plot, you will get the more information about that fields which belong to that particular part. So in the future direction, we are working with the international collaboration part, so that we can get more recognition about this for this IPD submission. And we are also working with the interactive environment to analyze the proteomics data, and also this in-house data visualization and validation tools for better consistency check when we get the data for the as a submission. And we are also actively participating in outreach activity to more provide the information about the what is happening at the IPD submission level. So this is the team which is working for this IPD portal. So this is me, my name is Pavan, and then software developer Rahul, and then Dr. Abhishek is a data curator, this Asa Verma is a data curator, and Neetu Kumari is working as a senior programmer. I want to acknowledge Professor Deepak T. Nair, our lead, and all IBDC core committee members. And I also want to acknowledge the RCB and DBT for providing this support. And if you want to you can use this web link to access the IPD portals. And if you find any query, then you can write to us at the IPD support@ibdc.rcb.ac.in. Now I will go to this simple live demonstration of this IPD portals. So, user can use this ibdc.dbtindia.gov.in/IPD to open this IPD portals. This is the home page. And you will get the information about the registration, submission, and you can browse the data and the submission process. You can click here, and this is the live dashboard. This information about the registered users and then submitted projects, and then the statistics. So first I will show the registration this login part. So I am using the test account. Once user login, the dashboard will look like look like this. So it will show highlight the number of projects user has created, how many projects have the metadata submitted information. If you have submitted any projects finally submitted, then it will count the number of submitted projects as well as the stages. So in which stage you have completed your projects this submission form. And from the if you want to create one more project, you can create here. So this is the page where user can click over the add projects and fill about the information about the project, any keywords, principal investigation name, PI email address, affiliation, and basic information about the funding details and project description. Once closed, then you can close it. Same way, you can fill the experimental detail about the projects. So first you have to drop down any available ID on which you will add the project information, and then about the how you have prepared the sample, then separate the peptides, then characterization of the peptides, and then information about the type of experimental workflow you have used. This means targeted, drop down, bottom up, or the some other type of workflow you have used during this characterization. Then instrument detail. So then it provides the information about this API, which fetch your required instrument name if you type some. Otherwise, you will get the all the list from the here. And what sort of protein information modification you have implemented, and some other information which is missing in that part of this, you can fill out here in this part. In metadata section, user can fill the information about the first select the project ID in which you want to add the project information. And then select the organism, then tissue, then cell type, and then disease. So the mandatory fields user have to fields, and all the information are connected to the standard API and the ontology information, so that user do not have to type anything. Still, if you have find anything which is not there, you can use this edit button option and change the information and update the form, so that you can again update as per your requirement. We will, once you use the update button, we will validate your information and standardize it and get back to you to change the information if any. Then at the last, user can go with this file submission, like either you have we have clearly mentioned, if you have 200 MB data to upload, you can use the web browser, or if you have larger than 200, then you can go with the FTP. So if you have some, so first it will ask for this type of submission you are providing. If you are providing this partial, then select the partial. Then FTP credential will open up. You can use this FileZilla to transfer. First log into your FTP account, then transfer all the files here. After transferring the files, you can go with this view and verify button, so that you can verify whatever you file number of files and you have uploaded in terms of MD5 checksum, so that transfer can be validated and number of files can be validated. And once you have validated this file, you can submit, and final button will come up to submit the data. If you move to the home dashboard, you can click over any project button, so that you will get the project detail about this project information that you have filled up during the project creation process, and then experimental part, then metadata part, that is the login profile. User can submit the data. Now just look at the browse data. If you look at the browse data, then it will list the number of all the deposited submission studies. And here, if you click over this any ID, then you will get the whole sort of information about this project, metadata information, experimental details. You can get this metadata fields and description in the PDF or the CSV format, or you can download the files related to your work. So just click over here and it will download the file in your area. User can further search the data. So using any keywords, just simple words. So all the matching words will be highlighted and the number of projects will be listed so that user can get the information about the protein related to your search term. And if you find an information project, then go to this IPD in the browse table and get the information from the browse table. At the last, this is the visualization part. So user can get information about this all the data information. So if you put over any detail, then you will get the tissue details, then cell type detail, disease type details like that. And at the last, you will get the number of files which are open access and different sort of files which are open access format. And this is the count number that you will get here from this statics page. So that's all about the proteomics data. If you have any questions, then we will go to that. So if you have any questions, you can ask now. If you do not have and still in future you got any query related to this IPD data submission, then you can reach to us at the IPD support at ibdc.rcb.ac.in, and we will communicate your query over the email. If any other information you want, you can write to us. So if we do not have, then we can close this session. And thank you everyone for joining.